autologous DNA and stem cell therapy. First, let me just tell you that stem cell itself, the word was coined only 20 years ago by 1998. Yeah, pause, 1 second. By 1998. Earlier to this, we gave them all nice names, multipotent cells, unipotent cells, cells which could do replication with very good duplicity, yeah? So this kind of a cell was called a stem cell only by 98. Earlier we called them multipotent cells, unipotent cells, and whatever potent cells, the totipotent cells, or whatever that we had. A lot of my work earlier was with different, there is a process called evolution. And this evolutionary thing actually runs in with each one of us. So I was actually, I need to just give you a 30-second point about, I'm just very, very lucky that I was born an eye surgeon. And I was born into my mother being a Muslim, my father being a Hindu, me not believing in any God, and me having only science as my background. Everything, you had to show it to me to prove it. Otherwise, I wasn't buying your theory. And this thing was so ingrained, and I'm guessing that all of you here have the same too. You have to see it. You can't just say, "Ah, bhai, chalta hai." No. You have to show me. You have to tell me. And then you have to show me again and again and again. Then I might believe what you're saying. And the same was true with cells. Now we are seeing the cells. Usually in ophthalmology, we're doing very, very minute surgery. So we're talking about micron surgery. And this is just very lucky that I already have an ultrasound in my hand by which we're removing cataract surgery, which I've been doing from the time I was 10, 15 years old, just because my parents and my grandfathers were all doctors. So it was very easy that I was inculcated in that science and in that medicine. That set me on a motion, like how we have taken you also through a process of evolution through this whole convention, and you saw Dr. Vyas giving you a beautiful talk on human energy. So I actually made presentations on magnetopexy at the International Congress of Ophthalmology right from 1994, 1989, and way back. So maybe 30, 40 years ago, I've been making presentations on this. From magnetopexy, I went to ultrasound, which I'm explaining to you, the cataract surgery is using an ultrasound in your fingertips. From there, I started using all those energy formats, devising all kinds of different machines, till I understood that cellular therapy has to come. Till then, and even at this point of time, medicine relies too much only on pharmacology. So we are relying only on chemistry and chemical molecules. We need to change that. We need to go to a biotechnological field. We need to make sure that different areas of our existence can come into play. What Dr. Vyas also was trying to explain to you was quantum physics. I didn't want to disturb him then, but I would like you to know now. The fact remains that in science, we ourselves still don't understand quantum physics. How are we going to explain how the medicine works on quantum physics? First, we should understand that. We don't understand. Our biggest physics experts have got half a dozen different theories on quantum physics. If you try to read quantum physics, at that point you will understand what I just tried to explain to you, that what my Guruji had been explaining to me coming out of the Vedic sciences is all quantum physics. We couldn't understand quantum physics. Where are we going to understand Vedic sciences? It's all one big world. We are not going to understand it. Let's not go to that. So there are certain areas that you're going to understand, certain areas that you're not going to understand, and certain areas that you will understand and you will play into practice. One of them became mana. So working on cell therapies, like I explained to you yesterday, I was trying to keep those fetal stem cells alive for a little longer time. Trying to do that devised and developed a culture medium. That culture medium now became known as manna. When I used it on fetal stem cells and on adult blood cells, I saw that they were becoming beautiful cells. At that point of time, I had no idea that this was a stem cell, but I did know that the cell is too beautiful. I started using that in practice. After doing more than 2,000 cases, I took the whole project. I said, I don't want anything to do with it. And that's when I met Dr. Kalam, and Dr. Kalam said, no, please go IPR, do this, and he gave me a whole big thing to do. Not Government of India, but DRDO. But DRDO is Government of India. And so they did a lot of my science. And one of those machines is what I'm going to show you. This machine is actually from Indian Institute of Science. So this is a very, very nice machine. It's an inverted microscope. It can take this, what you're seeing, that whole screen is actually less than 20 microns. And it can take 1 second, 125 frames. So it is so fast. See, when something is happening correct and fast, it happens very fast. So when your DNA is starting to form, it forms like this in second. Even 1 second is too much. What you're seeing is we have pulled that video, which is 125 frames. It's actually just 1 second. What we saw was that in front of our eyes, DNA ribbons were getting formed. And that's what you're going to see. Now, nobody can show you that kind of a video because 1, they don't have manna. 2, I don't have that machine. Okay, so when you play that video now, what we did here is we used an 0.2 micron filter in the process of making manna. We have to filter it with the 0.2 micron filter, whereby we take away all the big cells and all. Come in front, come in front. It's not possible. Come there. Come there. We have to filter away all the big protein molecules. So there's really nothing in manna. And on the other side, we are taking the second droplet in another syringe where we're taking a patient's blood. We have filtered that. We've taken the plasma. And then we're filtering that also. So here also we have taken off all the big protein molecules, all the cells, nothing there. When we mix both of them, we see those ribbons forming in front of us. Can you just rewind on that once more? Play that video again on that DNA. What you're going to see right now, see the fibrils forming in front of your eyes. That is the beauty of manna. And then the ribbons are getting laid down. As the fibrils form, the ribbons are getting laid down. And then we need to reconfirm that this is DNA. Because we have done this experiment thousands of times for decades, we know that it is DNA. However, to reconfirm it, there are particular stains that we use. Some of them are fluorescent stains, and that will show us the fluorescence when it is a DNA particle. Quite obviously, a small town eye surgeon doesn't have that kind of science. So it had to be with a communication and collaboration with all the biggest scientific experts of the world. DRDO, IISC, Max Planck in Munich are some of the major contributors, but there are independent laboratories in US of A. Every step of the way, once we thought, okay, DNA is getting formed. So we said, okay, DNA genesis, we're doing DNA genesis. And then we came to IISC, and IISC, there's a very, very emeritus professor, Padmanabhan. He told us, no, you are not DNA genesis. Don't put that onto your feather. That's not what you're doing. There is DNA in the blood of the patient, however minuscule it might be. That same DNA particle, which cannot be seen under the microscope, becomes vivid under the microscope when you form and place manna, because the DNA fibrils are now being formed. So what that actually means, if you're trying to make a building, you need to have the foundation laid very nice and strong. The stronger the foundation, the higher the floors. So if you have a weak foundation, you might just get a hut. If you have a strong foundation, you have an Eiffel Tower. So it depends upon the foundation. And that is the theory